this is a work that the leader of this project, I am not the only one, is Dr. Paschini, the leader, one of the leaders of this project, and with the same idea to introduce the transferrin by intranasal pathway. We use extracellular vesicle. The idea is to include in these vesicles the transferrin and induced by the remyelination using, again, the intranasal pathway. We still need to run the experiments to know if really the extracellular vesicle go through the intranasal pathway and reach the central nervous system to induce remyelination. Unfortunately, because of the pandemic, many of the labs were closed, and we are a little bit delayed in this kind of experiments. The experiment that we have published until now are the experiments that demonstrate that we can isolate the extracellular vesicle, that we can load the transferrin inside of the extracellular vesicle, and now we need to run first in vitro experiment to know we really go to the cells that produce myelin on one side, and the second part of the experiment will be we are able to use the intranasal pathway to reach the central nervous system with this vesicle that will be low with transferrin. The idea to use extracellular vesicle is that the transferrin is protected by the degradation when you use by any pathway, and you have a more clear control of the amount of transferrin that you can use to induce your remyelination if really in this experiment we are successful. The critical point here is how this vesicle can reach specifically the cells that produce myelin. So these are the oligodendrocytes. In this way, we are trying to develop different strategies, but this experiment needs to be done yet.